So the next thing on our what we need to know list states that we have to know the difference between RNA and DNA. And DNA versus RNA. Well, you already know that the D in DNA stands for deoxyribose. So DNA has deoxyribose, RNA has ribose. So they differ in their pentose sugar. DNA has adenine, guanine, cytosine, and thymine. RNA has adenine, guanine, cytosine, and uracil. Okay, so if you ever see a thymine, you know you're dealing with DNA. If you ever see a uracil, you know you're dealing with RNA. Okay, DNA happens to be double-stranded, and we're going to talk about the hydrogen bonding between the complementary base pairs. RNA, on the other hand, is single-stranded, so RNA doesn't have a double strand, just one single strand. DNA is usually found in the nucleus, unless you find some DNA in the mitochondria, but we're mainly focusing on the chromosomes in the nucleus, and the chromosomes are made up of DNA with protein wrapped around it. The protein these are called histone proteins, and the histone proteins are attracted and wrapped around the strand of the double-stranded DNA to make a chromosome. So they're found in the nucleus. RNA, on the other hand, there are several types of RNA. Some RNAs are found in the nucleus, some are found in the cytoplasm, at the ribosomes where the proteins are being synthesized. So you find lots of RNA in several different places. DNA is just made up of thousands and thousands and thousands of nucleotides, double-stranded, complementary base paired. RNA, you have some long RNAs and some short RNAs. And there are lots of different types, and we'll talk about the different types of RNAs. DNA has genes. So these genes are what code for specific polypeptides, specific proteins, or portions of genes are there to code for a portion of a specific polypeptide. So the genes are found in DNA. The RNA, you find the exons and the introns and whatever is going to be decoded by the genetic code to create the primary structure of a protein. All right, let's focus now on the different types of RNAs. So this is my list here. Let's focus on the different types of RNAs. There's, and we'll be mentioning them as we talk about transcription and translation. So types of RNA. There's heteronuclear RNA. There's small nuclear RNA. There's mRNA. There's rRNA. And there's tRNA. And we'll be seeing all of these as we discuss transcription and translation. All right, but before we get into all of that, let's go ahead and talk about the complementary base pairs. How is it that a DNA molecule is double-stranded? And I just mentioned that there's hydrogen bonding between the complementary base pairs. What are the complementary base pairs? Well, adenine will always complementary base pair 2 hydrogen bonds with a thymine. So if one strand of DNA has an adenine on this side, the opposite side on the other strand, there will always be a thymine. Okay? And there are 2 hydrogen bonds. Anytime you have a guanine on one strand, you'll always find a cytosine hydrogen bonded with it on the other strand. And there are 3 hydrogen bonds. Okay? Now, how does that actually look? Well, let me go ahead and draw out an adenine. Here's an adenine. Well, think of it as an adenosine because it's part of a long It's a nucleotide that's hooked up to thousands of other nucleotides. But I'm just going to show you just a plain old adenine, how it would hydrogen bond, all right? So again, if we're talking about a DNA molecule, okay, if there's an adenine here, there's going to be a thymine here, and you get the hydrogen bonding, okay? But right now, I'm just going to do an adenine by itself and show you how hydrogen bonds with thymine. I'm going to flip the thymine over because anytime you've got a DNA double strand, this one strand goes 5', 3', and then the opposite strand, the one that's complementary base pairing with it, is 3', 5'. So they always go in opposite directions. So I'm going to just flip this thymine over just so that you can see what it would look like with the hydrogen bonding. Remember, thymine's got 2 carbonyls. It's got a methyl group over here. All right. So remember, these are hooked up to the sugars, and then the sugar to the phosphate, and you get the sugar-phosphate backbone. And really, in order to do this justice, so think of these lines as the sugar-phosphate backbone. This is an adenine, this is a thymine, and they're hydrogen bonding with each other. So the lines are sugar-phosphate backbone, and then you've got these projections. So maybe you have a guanine here, cytosine here, and that's the hydrogen bonding. All right, so let's take a look at the 2 hydrogen bonds that are going to occur between an adenine and a thymine. All right, so this nitrogen has a lone pair, this nitrogen lone pair. Okay, all the nitrogens have got some lone pairs there. All right, see this hydrogen that's attached to this nitrogen is going to be attracted to the lone pair of this oxygen. So we can squish them together. That's one of the hydrogen bonds. This hydrogen on this nitrogen of the thymine is attracted to this lone pair. Those are the 2 hydrogen bonds that occur between adenine and thymine. No other hydrogen bonds, so nothing over no place else. These are the only 2 hydrogen bonds. Okay, now let's take a look at guanine and cytosine, what they look like. So I'm going to draw my guanine over here. Just going to do a plain old guanine here. I have a carbonyl here, an NH2 here, right? This has a hydrogen. So this is guanine. This is my G. All right, now I'm going to flip over my cytosine so that we can show the actual hydrogen bonding here. Erase this a little bit. Actually, let me get a little closer. I'm stuck. I have to erase this so I can show it properly. All right, so I'm erasing this. All right, so this is going to be an NH2, and the carbonyl comes here, and this. There we go. So this is my cytosine. This is my guanine. I'm going to put my lone pairs where they're supposed to go. Lone pair, lone pair, just so that you can, I mean, honestly, I don't care about the others. I'm just focusing on where the hydrogen bonding is. So 3 hydrogen bonds between guanine and cytosine. So see this hydrogen on this NH2? It's going to be attracted to this lone pair over here. See this hydrogen on this NH right over here? Going to be attracted to this lone pair. And see this hydrogen over here? Oxygen has lone pair. There you go. Here are the 3 hydrogen bonds. Okay? So if you have a stretch of DNA, okay, 5', 3', and let's say the stretch is A, G, C, C, T, G, A, T. On the complementary base pair over here on the second strand, it's going to be T, C, G, G, A, C, T, A. And then it goes in the opposite direction. This is 5 prime and this is 3 prime. Okay? So now that we know what they all look like, we're ready to move into replication, transcription, and translation. And that's what we will do next in the next video.